And now, on to our next session, brought to us by Danaher Life Sciences, Enabling Science at Scale, presented by Vanessa Almendro, PhD, Vice President of Science and Technology, Head of Innovation at Danaher Corporation. Vanessa, over to you.
Hello, everyone. It's a real pleasure to be here today. My name is Vanessa Almendro, and I am the Head of Innovation at Danaher Corporation. Today, I'm going to tell you about Danaher Corporation and how, with the portfolio of technologies from our life sciences companies, we can create workflow solutions for your R&D and manufacturing needs. And I will end my presentation with a couple of specific examples on how we can use integrated workflow solutions for cell line development and mRNA manufacturing. While Danaher might not be familiar to some of you, I am sure that most of you are very familiar with our brands. In fact, Danaher is a science and technology organization that is focused on life sciences and diagnostics. Our life science organization is comprised by brands like Beckman Coulter, Leica Microsystems, Molecular Devices, SCIEX, IDBS, Phenomenex, Aldevron, Cytiva, and many others. Some of our diagnostic brands are represented by companies like Cepheid, Beckman Coulter Diagnostics, Leica Biosystems, and Radiometer. And for those of you that do know Danaher, you also know that we have another area in our business, a subset of portfolio of companies that are focused on environmental and applied sciences. This year, we announced that we are going to spin off this part of the portfolio as an independent publicly traded company named Veralto by the end of the year, which is going to be leaving Danaher as a fully focused science and technology organization. And at Danaher, we have a true commitment to innovation that can impact human health outcomes. And we apply the power of science and technology to create innovative solutions that can improve health outcomes through our proprietary combination of our people, our capital, and the Danaher business system. And when we don't develop drugs, the Danaher family of operating companies, together with the extensive portfolio of technologies and services that we have, with our culture of continuous improvement and operational excellence, with our multidisciplinary innovation capabilities, collectively, we aim to accelerate the positive impact of science and technology in human health. And we are truly committed to enable our partners and customers to deliver life-changing therapies more quickly to more patients and with the best quality. And with our portfolio of products, we can support not only the development of one therapeutic modality, but actually we can support multiple therapeutic modalities. And why is this important? Well, for those of you that have been in the pharmaceutical industry for more than 10 years, you have witnessed that we went from basically having only small molecules and antibodies to develop therapeutics to now have a plethora of additional modalities that are allowing us to advance the preclinical and the clinical research, to advance life-saving therapies for many more patients than ever before. But as we have developed this significant armamentarium of therapeutic modalities, the complexity on the manufacturing process has also increased. And as manufacturing processes have become increasingly complex for modalities like, for example, cell and gene therapies, maintaining the control on the manufacturing and achieving consistent results can be more difficult, and it can lead to operational problems, including quality issues. And this is exactly what we can do at Danaher with our extensive expertise in multiple technologies across multiple therapeutic modalities. We can help our customers to understand this complexity and to streamline research, development, and manufacturing. And in fact, Danaher's commitment to innovation and to patient impact has been instrumental to advance the biomedical field. And in this slide, I just have a few examples on how we have responded to biomedical and biomanufacturing needs. For example, during the COVID response, Cepheid gold standard point-of-care molecular test was the first to market COVID-19 diagnostic test. Our companies were instrumental in responding to the surge demand of synthetic nucleic acids, and IDT was the first company to get their primers approved by the CDC to be used in COVID testing. In addition, our companies quickly pivot to provide dedicated manufacturing capacity for both diagnostics and vaccine biomanufacturing. And between Cytiva, Beckman Coulter Life Sciences, and Molecular Devices, we supported more than 400 COVID-19 vaccine and therapeutic projects, including all the Operation Warp Speed candidates. Similarly, in the genomic medicine space, our life sciences companies, including Aldevron, Cytiva, SciEx, and Beckman Coulter Life Sciences, just to name a few, have played a critical role in advancing time to market of cell and gene therapies. For example, we created many of the technologies, processes, and products that supported 4 out of the 6 currently approved CAR-T therapies. We also supported the manufacturing of CRISPR-Cas9 enzymes that are now being used in multiple clinical trials. And building on these past successes, Danaher is now doubling down in our commitment to science and technology and innovation. And during the past year, we strengthened our scientific leadership by bringing several additional industry veterans. Now, under Jose Carlos Gutierrez Ramos' leadership, and with more than $1 billion invested in research, we are upscaling our innovation efforts to continue to create solutions that can help our scientists to interrogate biology faster and to develop therapeutics with the appropriate quality, speed, and scale. And in particular in life sciences, as I mentioned before, our goal is to partner and support our customers with technologies and solutions that reduce research development and manufacturing costs and make a meaningful impact in accelerating time to market, which ultimately is going to impact their core growth. With our global presence and our more than 36,000 employees worldwide, and this is only for the life sciences business, we deploy a broad portfolio of products that are helping both public and private institutions to advance biomanufacturing worldwide. And as your partner, there are 3 things that I would like for you to keep in mind as you are thinking about your drug development needs. First of all, when you interact with one of our brands, you are in fact accessing Danaher as a whole. Point number 2, in working with Danaher, you are accessing our global insights, and we can help to challenge outdated assumptions and proactively bring new ideas to the table to help you to innovate faster, which is, of course, critically important in this fast-paced industry. And lastly, also critical and important, by being a global company, we can leverage our extensive service and infrastructure to support you now and as well as you grow and expand your business. In this slide, I have some particular examples of the extensive portfolio of analytical solutions that we can provide to support the development of your drug candidates. And we understand what are your pain points. As I mentioned before, most of us and part of our leadership are coming from the drug development space. And we understand really how challenging it is to develop drugs. And with technologies like microplate readers, mass spectrometry, particle analyzers, separation reagents, data support, and many others, what we want is to have you to get the right analytical tools that are necessary to ensure the reproducibility, precision, and sensitivity of your processes. To just cherry pick a few examples, the IntaBio CT from SciEx provides an integrated solution for protein analysis that includes automated sample preparation and data analysis. The BioPhase 8800 capillary electrophoresis, also from SciEx, has a standard work that allows for parallel processing of 8 samples at a time. Another example is our Optima AUC analytical ultracentrifuge from Beckman Coulter that is broadly used to analyze partial, full, and empty capsids during viral vector manufacturing. And let's not forget that in addition to this, we can also support your RUO, GMP, and drug product manufacturing needs with companies like IDT, PNI, Aldevron, and Cytiva. And in addition to these point solutions, Something that might be even more valuable considering the complexity of biomanufacturing is our ability to take all these technologies and integrate them to provide workflow solutions that can help you to build not just new capabilities, but also to implement optimizations in your existing capabilities. We have a fantastic team of experts and solution architects that can work side by side with the leaders of R&D and provide this support at any stage of drug development and for multiple therapeutic modalities, including cell and gene therapies, mRNA therapeutics, and biologics. And now, let me just move towards a couple of specific examples on how we have taken these technologies and how we have integrated them to provide workflow solutions. And I'm going to start with the biologic manufacturing. During the past 2 decades, we have seen a phenomenal evolution of the monoclonal, bispecific, and even the trispecific antibodies for therapeutic applications, with the monoclonal antibody global market on its own being more than $200 billion. Within each type of monoclonal, bispecific, and trispecific antibody, there are different target product profiles that can be generated depending on how the different antigen binding moieties and the other components are combined. And these different TPPs will have different developability considerations for stability, solubility, and other parameters that confer drug properties. And the versatility in the way that we can design biologics offers a tremendous opportunity from a therapeutic perspective, but it also poses multiple challenges from a development perspective, since there is no one-size-fits-all solution, and each desired feature of the final product mandates the need to have a diverse panel of assays and technologies. And to ensure the production of these high-quality biologics, drug developers need to optimize upstream manufacturing challenges like cell line development. In fact, cell line development is one of the most important rate-limiting steps in bioprocessing for biologics manufacturing. Cell line development is a multi-step, complex process that needs to be carefully optimized since the vector design and production to the final stage of cryopreservation of the final cell line bank. And as we have been talking with our customers, we have learned that there are many specific pain points in cell line development, including stability and expression level, achieving, for example, the desired functionality while maintaining the high expression levels that are desired can be challenging just because of the complexity of the way that we engineer these proteins. Immunogenicity. We know that there is a risk for immunogenic responses, and there is the need to have the appropriate analytical methods for product characterization to select the right master cell bank. Product quality and scalability are also critical. The antibody production can be heterogeneous, which can lead to variability in the protein expression, assembly, purification, which can ultimately impact the final product quality. And again, having the right analytical methods to understand the critical quality attributes early on is really important to select the master cell bank. In addition to this, of course, data complexity is a challenge in cell line manufacturing, given the multiple analytical systems, the traceability, and the accurate documentation of the experimental parameters that needs to be monitored throughout the process. To overcome some of these challenges, Beckman Coulter Life Sciences, SCIEX, and IDBS, for example, are currently offering several solutions. And in this slide, I just want to highlight 3 technologies that we believe can help in accelerating cell line development. For example, technologies from Beckman Coulter Life Sciences and Molecular Devices can accelerate the time in the screening process. The Valita titer helps to screen cell lines for IgG production earlier in the process, only in minutes, not in days. The ChemStress can allow to predict which clones and media will be best for scaling up. It can help you to understand the scale-up properties of the clones. And it is not in this slide, but Molecular Devices also has several instruments like the clone peaks, that automates cell culture and monoclonality verification, which helps in avoiding the need to have to capture the image, one by one of the cell cultures over a period of time. The IntaBio CT from SciEx, that, as I mentioned before, that can provide an integrated solution for protein analysis that includes automation, sample prep, and data analysis. An IDBS Polar can help in removing the tedious manual data process and help to capture the data in an automated way, providing a backbone for data process, improving accessibility across the process, and improving access for everyone. And these are just, again, a few examples that I wanted to highlight about our analytical solutions. Of course, with the breadth of technologies that we have, what we can do is to help you to overcome challenges also in automation, data management, and as I mentioned before, we can help in providing full integrated services. And another example that I would like to highlight on how we can support your end-to-end manufacturing needs is on mRNA. While the potential of mRNA was discovered more than 30 years ago, of course, after the success of the mRNA technology for COVID vaccine applications, we have seen a booming of the field and an expansion in the use of this modality in multiple additional applications. mRNA is expected to become a $3 billion market by 2026, with more than the 150 therapies that are right now in the clinical pipeline. The increased speed from discovery to delivery makes mRNA a very attractive modality for vaccines and protein replacement therapies. Many of the challenges that are associated with the development and manufacturing of mRNA are actually reminiscent of the early days of the monoclonal antibody development, where the industry was struggling to overcome the low titer and the poor production yields challenges, which was leading to very costly and inefficient commercial manufacturing. As the industry focused the efforts to overcome all these production challenges, that resulted in the monoclonal antibodies becoming the fastest growing class of biopharmaceutical products. The mRNA manufacturing process is made up of discrete, specialized steps, highly dependent on the size of the construct, the capping strategy, and the IVT efficiency. And there are many challenges associated with these processes. One of the main challenges is, as well, supply chain instability, especially when using multiple vendors for such individualized steps. Not only our customers have seen significant issues in delays with raw materials, but also the handover of the product between suppliers can impact the time, the cost, and of course, the quality of the final product. In addition, there are challenges associated with the raw material variability that can also significantly impact the impurity profile of the mRNA molecules, which will lead to the requirement of different purification steps from case to case. And there are many other critical quality attributes like capping, the poly-A tail stability, and many other qualities similar to the antibodies that need to be overcome. And similar to the antibodies as well, the data management can be complex due again to the complexity of the manufacturing process. And similar, as I mentioned before, for monoclonal antibodies, we are able to provide solutions to our customers, both for those that desire to outsource this complex manufacturing process for mRNA, as well as for those that want to insource the manufacturing process. For those that desire to outsource the manufacturing process, with Aldevron, we see a way to reduce the mRNA drug product time to market up to 40% and hand the entire manufacturing process for our customers from the sequence to the vial in a single location, just with a single vendor and under a single quality management system, which can provide significant benefits. In addition, for the clients that desire to insource the manufacturing of mRNA, Cytiva can provide equipped labs to clients, and a set-up manufacturing setting in order to retain this internal control of the later stage and commercial products. So that can help in avoiding having to deal with the challenge of outsourcing the simple steps to multiple providers. So to summarize, Danaher is a science and technology organization that is focused primarily in improving human health outcomes. And we are doing so by maximizing the research and the investment that we do in human systems R&D, as we are trying to support many more therapies that can be discovered and commercialized faster. Another area of focus is genomic medicine. I provided a few examples, but we are here to enable novel genomic therapies that can deliver cures faster. We, of course, are very focused on developing technologies, again, that can help us to not only develop the therapeutics, but to identify the patients that can benefit from these treatments. And we are revamping right now our strategy in diagnostics to be able to expand our portfolio of assets to develop diagnostics to treat the right patient at the right time. Needless to say, with our companies like Cytiva, we are leaders in the space in developing the technologies that enable next generation biomanufacturing. And behind everything we do, there is a significant amount, of course, of data that we are using to learn and improve our processes so that we can develop the best solutions to our customers and partners. If you want to learn more about Danaher, please visit our website. I really appreciate all your time and efforts. I'm going to leave you next with a video about Danaher, and I will be following up with all of you with your questions. Thank you so much.
As the life sciences companies at Danaher, we see a way to empower our customers to bend the drug development time curve by 50%. To unite science and industrial engineering, to eliminate bottlenecks and pain points, while innovating to improve your entire R&D pathway.
We see a way to help you deliver novel, high-value therapies faster than ever before, to the benefit of patients around the world.
We see a way to accelerate what you can accomplish every day. By partnering to automate, digitize, and scale your workflows, we develop solutions no one has seen before, driving the kind of outcomes no one has achieved before.
How can we see it? Because we have the outcomes to prove it.
Together, we see a way to empower our customers to reduce time to market of life-changing therapies by 50%.